Good morning, student. We are going to discuss electrophoresis, the topic I was already started in class. So those who are present in the class, I have already given introduction and the principle. I have defined how to measure the velocity of the electrophoreting particle and then the factors affecting electrophoresis. So today we will start with different types of electrophoresis. We will start with the concept of gel and then the electrophoresis of gel electrophoresis, different types of gel electrophoresis. We will start with the principle. Electrophoresis is the migration of charged particle or molecules in a medium under the influence of an applied electric field. So typically we are seeing here a chamber where the two electrodes are there, which is already linked with the power supply. The charged molecules are there in a solution. Once the power supply is on, they will be migrating, depends on its charge. Positive charge will migrate to the negative electrode, negatively charged particle will move to the positive electrode. The relative speed depends on their charge, size, shape of the molecule, and you have already studied how the different factor can influence this migration. Now, I discussed about the history of electrophoresis. The discoveries are there. Initially, the electrophoresis scientists, they have carried out in free electrophoresis. There was no solid support. So generally in a solution, they have checked the movement of blood cells, bacteria, all these, and they have measured also those movement. Now they understood there's a requirement of a solid support matrix. Now what are the extra support this matrix are giving? It inhibits diffusion fast. The major problem was the diffusion, and because of the diffusion, generation of convection current, which would otherwise impede separation of molecule. The solid support matrix, it allows a permanent record of results through staining after running. So this documentation is very much important, and it can provide additional separation through molecular sieving. Now if it is the solid support itself can help the separation due to its sieving property. We will see what is the sieving property. Now we'll start with what is a gel. Gel is a cross-linked polymer whose composition and porosity is chosen based on specific weight and porosity of the target molecules. That means it's a type of a cross-linked polymer. We will allow them to cross-link in such a way that they will form some pore, and through that one, the molecule which you want to separate, you are allowing them to flow, and they are generally inert and they will not interact or do any reaction with the molecule we are going to separate. And because of that, the molecule will separate based on their the charge, their molecular weight, based on this property. So the types of gels commonly used in laboratory are starch gel, agarose gel, polyacrylamide gels. So there was a time when agar also used, but now it's not so popular. Starch gel was one time as very popular in order to separate the blood protein and some isoenzymes also, and people have done their activity study also by using starch gel. Starch can be hydrolyzed. Potato starch can be used in acidic condition and then can allowed to solidify in between two glass plate, and can be used as a support matrix for separation of the molecule. Now the problem was with the starch gel is that it's get contaminated. Bacterial contamination, fungus contamination was very much there. So nowadays seldom use this type of gel. Now, the popular one is agarose gel for separation of nucleic acid and some cases protein. We will see for the only nucleic acid, and polyacrylamide gel for separation of most likely for the proteins and small nucleic acid. So we'll see in details what is this agarose gel, polyacrylamide gel. Now, if the question comes like write the principle, I mean the agarose gel electrophoresis, generally you have to start with the principle, procedure, instrumentation, then detection, then application. So we will go like that. Some more information also we will be discussing. So gel electrophoresis, this is agarose gel electrophoresis basic apparatus. We are showing gel electrophoresis used a cross-linked polymers. Depending on what percentage of gel we are using and and what molecule we are going to separate, that is also another criteria. Based upon we will choose the porosity. We can control its porosity. So this is a typical a tray is showing where the glass plate, on top of that you have casted the gel. It's a submerged kind of gel, and it is submerged inside a buffer solution, and the electrodes are there which is linked to a power supply, and the whole thing should be covered. Okay, so something about the technique of electrophoresis, instrumentation and the reagent what is required. So we are seeing the two buffer boxes here, two chamber you are seeing. Using the buffer used for this process. The buffer generally we use for electrophoresis in agarose gel, that is a TAE buffer or TBE buffer. And the two buffer each buffer box contain an electrode made with either platinum or carbon, the polarity of which is determined by the mode of concentration of the power supply. So which plug you have plugged in, based on that only, whether it is a anode or cathode. The electrophoresis support on which separation takes place may contact the buffer directly or by means of wicks. So generally nowadays we will see that it directly connected. The entire apparatus is covered to minimize the evaporation and the protect the system. So we have discussed already the what is the effect of temperature of electrophoresis. More temperature, it will evaporate, and most of the cases the water molecule from the buffer will be evaporated. The ions will be concentrated. The ionic strength of the solution of the buffer will change, so it will run much more faster, leaving your sample behind, so the incomplete separation will take place. So maintaining the temperature so that the electrophoresis separation will be very correct. It's very much important point. The power supply to provide electric power. So these are the things required for instrumentation. Some basic common things of agarose and polyacrylamide, even though I will discuss agarose and then polyacrylamide. Although agarose and polyacrylamide differ greatly in their physical and chemical structure, they both make porous gel. So gels are porous. So the porous gel act as a, remember this word, sieve, by retarding or in some cases by completely obstructing the movement of macromolecules by allowing the smaller molecules to migrate freely. So you'll see sometimes that we are not depends on the charge of the molecule, only the molecular weight is the factor. So the bigger molecule, because of this shape and size, will be retarded because the pores are not big enough to allow them to flow, but the smaller molecule, instead of that, they will migrate. By preparing a gel with a restricted pore size, the operator can take advantage of molecular size differences among the proteins. So when we will discuss the protein separation by polyacrylamide gel, it will be much more clear. So it will only depends on the molecular weight. If I somehow make the protein charge same for all the protein. Now again, because the pores of an agarose gel are large, agarose are used to separate macromolecules such as nucleic acid, large proteins and proteins complexes. Polyacrylamide, which makes a small pore gel, is used to separate most protein and small oligonucleotides. That means if it is a protein is very small, it's not good enough to separate by agarose gel. Generally agarose gel we use for nucleic acid and polyacrylamide gel we use for protein separation. Remember, both are relatively electrically neutral and they generally don't interact with the proteins, neither proteins nor nucleic acid. Agarose is a highly purified uncharged polysaccharide derived from agar. Agarose dissolved when added to boiling liquids, that means buffer. It remains in a liquid state until the temperature lowered to about 40 degrees centigrade, at which point it gels. Its gelling properties start once you boil and then allow to cool them. Then only its forms its gelling property. Otherwise, it comes as a powder. The pore size may be predetermined by adjusting the concentration of agarose in the gel. Agarose gel are fragile, however, they are actually hydrocolloids. They are held together by the formation of weak hydrogen and hydrophobic bonds. Remember, there is intramolecular and intermolecular hydrogen bond. So, what is actually the agarose? It's a repeating unit of actually galactose, beta D galactose and 3,6 anhydro L galactose. So if you can remember, see, it is GA, repeating units of galactose and anhydrogalactose. So that is 3,6 anhydrogalactose. Can you, if you remember, fine. If you can able to draw the structure, then only go for it. Otherwise, don't do in exam. Now see the gel structure of agarose, what actually informed here. So this is a sol state, 45 degree, the initial gel. Then aging, it forms final gel structure. It will be much more clear if I show you this one, and hopefully when you are writing the principle of agarose gel, you should mention this thing. There's an intramolecular hydrogen bonds agarose polymer. Okay, once you heat, it gets denatured. Okay, then again you are cooling. So everything you are doing, mixing the agarose powder with the buffer, certain percentage, like if you want to prepare 2% agarose in a 100 ml solution, so 100 ml buffer, and you are weighing the 2%, 2 gram of agarose powder, and then you are heating it 70 to 80 degrees centigrade. It should boil and give you a clear solution. And then what happened during this clear solution formation? It's actually denatured. And then when you are cooling, intramolecular hydrogen bonding, so intramolecular and here intermolecular hydrogen bond formation takes place. See the how the gel apparatus, horizontal or submarine gel apparatus looks like. This is kind of a complete trough where this is the place where you can keep your gel. These are the two tank. And this is the way you are actually loading your sample, and this is the way you can turn on the power supply and electrophoresis can be done. Now detection. Now detect bands by staining during or after the electrophoresis. Generally we mix ethidium bromide for double-stranded DNA. It gives a pink color, typical pink color under UV light, because ethidium bromide is an intercalating reagent which actually go inside the double strand of DNA and can get and ethidium and can give you a fluorescence pink color under UV light. Cyber green or cyber gold for single or double stranded DNA and RNA can be used. We can use silver stain, more sensitive for single or double stranded DNA or RNA. So Kumasi blue also can be used. So you can see that there are different type of dyes are available: amido black, Kumasi blue, Cipro orange, nail red. These are used for the protein, and polysaccharide can also be stained with iodine. So see the detection. The first one is the ethidium bromide stained DNA fragment. This is a silver stained. This is cyber green. This is Kumasi blue. This is again silver stain. Application of agarose gel electrophoresis. Estimation of the size of DNA molecule following restriction enzyme digestion, that is restriction mapping of a clone DNA we can do. So you can remember the restriction mapping. You can, after digestion with the restriction enzyme, you can just run the gel, you can see the fragment size. Analysis of PCR fragment, that is the molecular genetic diagnosis or genetic fingerprinting. Separation of DNA for extraction and purification and cloning. Separation of restricted genomic DNA prior to southern transfer or RNA prior to northern blot. So this is usually regular practice before doing an southern blot, northern blot. We need to cut the DNA with the restriction enzyme, and followed by we do an electrophoresis. So I will this is my first video. I will discuss the another topic, that is pulse field gel electrophoresis, in my next lecture. Thank you.